Yeah, circle Station 4, not to be confused with attorneys. This organism is a member. I don't want to insult any leeches. That's a member of the class Hyridinia, and the class lacks a couple things. It lacks internal septi. It lacks parapodia. It lacks key. So you could have said any of those, but they do have oral and anal suckers, which the other worms don't have. Station 5, that's a member of the class Polyplecophora, the chitin, and the pin A was in the foot, and the structures in the groove of B are cnidia. Station 6, the slug, the ant slug, those are members of the phylum Mollusca and the class Gastropoda, and the pin A is in the neustome is the best answer. Some people wrote the lung opening. I think I accepted lung opening. I think one person wrote mental cavity opening. So I'm like, okay, sure. You wore me down. So anyway, neustome's the best answer though. But I took some alternative answers. Mental cavity or lung opening. Station 7. This shell was produced by the, this is the clamshell. This shell was produced by the mantle, a muscular secreting shell gland. Label A indicates the umbo, and label B is pointing to the remnant of the, and I'll pull this shell out. I couldn't remember which I had because I was home grading these. So I had to come back and check it this morning. So B is in the, this is the posterior adductor muscle, ADD. Adductor muscle, and that would mean this is the left shell. So that's the that's how that works. That was the left shell and the posterior adductor muscle. So I know that's nitpicky. I think if you wrote adductor muscle or if you wrote ADD, adductor, not abductor, ADD, adductor muscle, or if you wrote the wrong one, the right one, the anterior one, I gave you half. And then that's the left shell, so that should be the left circle. Right? Or left. Okay. All right. So, station 8. That was the big clam in the jar. Label A was the foot. Label B is the siphon. And the feeding mechanism is a filter feeder. And clams are bivalvia. Station 9 is this was the nautilus and the cuttlefish bone. These are the phylum Mollusca and the class Cephalopoda. And the function of specimen B is mostly for buoyancy. It's that light pumice-like material that helps the cuttlefish float. Is specifically why I was asking for B. Station 10. This is the squid, whole squid. Pin A is in the siphon, and that is the posterior side of the organism. And pin B is in the mantle. The mantle is basically just kind of a large upper structure. Here's the squid dissection. Pin A is in the cnidia, and the hemolymph is supplied by a branchial heart. And pin B is at the organism's rectum or anus, where the ink sac exits the body. Station 12, that is the subphylum Myriapoda. The specimen in dish A is the centipede Chilopoda, and dish B is the millipede Diplopoda. Station 13, those are remnants of the, that was the barnacles stuck to the oyster shell. Those are in the phylum Arthropoda, and the class should be Crustacea, and they feed by filter feeding. The little crustaceans stuck to the clamshell. Station 14, the horseshoe crab, the anterior segment's called a prosoma, the posterior segment's called the opistosoma, and the horseshoe crabs are members of the class Xiphosaura, with an X. Station 15, the crayfish is a member of the class Crustacea. The pincer leg is called a cheliped, And the paddle tail leg is called a uropod. And that was a male crayfish. We have the claspers. Station 16, crayfish dissection. The A was in the cardiac stomach, or gastric mill was okay. B was in the pyloric stomach. And then C, I intended it to be the pericardium, but some people wrote the sand vein, and I went ahead and said, okay, fine. But it was in the pericardium, the main thing. Sand vein.
Yeah, it was kind of in the back of it, I think.
Okay, station 17, the scorpion is a member of the subphylum Chelicerata, and the pincers are actually modified pedipalps, the second appendage of all members of the group. The structure of pin A is called the pecten, or pectens, a unique sensory appendage, and the slits at pin B are the book lungs. I think I accepted. I don't know why I accepted it, but some people wrote book gills, and I didn't write it wrong. But book lungs is the best answer. Station 18. The tarantula is a member of the class Arachnida, and the fangs are a modified chelicerae, and the web spinners are remnants of the telson. Station 19. The cockroach organ labeled A should have been the crop. B was at the cecum, but if you wrote trachea rather than cecum, I think I accepted it, because it was kind of hard. I couldn't really stick it in the cecum because the cecum was too small. So it was next to the cecum, but there was also trachea there too. So if you wrote cecum or trachea, that was fine. The label C for sure was the Malpighian tubes, because it was right at the junction, and they fit like that. Station 20 is, those were trilobites. The subphylum is Trilobita. So just as a note, if I put two lines under the first letter of a word, it means you're supposed to capitalize it. So all these phylogenetic names are proper nouns. So Trilobita, for example, should be capitalized. Just as a note. Just kind of help you out. And the grouping body segments by function is called tagmosis. Okay, the last page. The frog's small intestines. This was rough. I don't know if it was because it was the lab right before we took our test, or if people didn't really study, or if it was confusing or what. But I tried to tell you what was going to be on the test, and I was trying to be very clear about that, and tell you what tissues I could possibly ask. So for the frog's small intestines, the pointer was identifying the simple columnar epithelium that was lining the inside. And the 100 on the micrometer scale should have been on the smooth muscle. Now it got bumped, but it didn't really get bumped to the far outside. I mean, it got bumped at one point in time, but it was still kind of like right at the edge of the smooth muscle versus the outside would have been stratified epithelium. So anyway, I had a hard time, accepting stratified epithelium, considering the monkey ear had a stratified epithelium as its answer too, right? So anyway, you can tell me yours got bumped or whatever, but I tried to put the 100 right in the middle of that smooth muscle band. That's the idea. Station 22, the cells at the pointer. Oh, this was striated muscle or skeletal muscle, and the function is for movement in animals. Some people wrote some alternative answers, and I might have given you partial credit. And that's the case with station 23. I put a neuron on there. This is nerve tissue. I thought it was pretty clear this was a neuron in neuroglia, but some people wrote that it was areolar connective tissue. And I'm like, kind of looks like it, doesn't it? So anyway, I believe if you wrote, okay, neuron is the answer that I intended, and then the two cell types were, the tissue is called nervous tissue, and the two tissue types are a neuron and neuroglia. But if you wrote the tissue being areolar connective tissue, and then you wrote the two cell types being matrix versus fibers, I think I accepted that. In general, I think I didn't even grade the two cell types because there were so many wide variety of answers. I began to feel like it didn't quite go over very well when we covered that. So, pretty much if you gave me any reasonable tissue type in 23, nervous tissue was the best answer. But if you wrote aerial or temporal tissue, I think one person wrote adipose tissue. I'm not sure if I marked it wrong or right. But anyway, so I just kind of realized, oh shoot, probably had too many labs doing it for this test. Maybe we should have pushed that lab off one time or something. I don't know. Everyone was at saturation. Okay, and then the monkey ear, I told you that would be on there. And the outer layer, which is pretty obvious because I say outer layer, is stratified epithelium. And it's separated from the inner layer by the basement membrane. And it says what structure is separating the tissues above from tissues below. It's a basement membrane. The inner layer at the 100 is cartilage. Specifically, it was elastic cartilage, but it's cartilage. It's the tissue there. Okay? Now, so there's a lot of variation in some of the answers here. So, you know, I tried to give you the benefit of the doubt on some of these. But I told you at the beginning that with epithelium, you have to say the full name, like simple columnar epithelium or you know, stratified epithelium. I allowed you to say stratified epithelium. Okay, so the dichotomous key we had over here at 25 was different. That was a true fly, right? 26, that was a tricky one. That was a Neuroptera. It was an antlion though. And a lot of people were close and they put Isoptera because of the big wings. But this one had no cirrus and it had long antennae, a couple other features, but it was a Neuroptera. Station 27 was a Hymenoptera. That was a little big monitored wasp. And you had to look carefully to see the two wings were two pairs of wings were there. And the mouth parts were mandibles. And then station 28 was a beetle, Coleoptera, that looked a lot like a red cockroach, but it was Coleoptera. And that one was clear because it had the T-wing. The wings were parallel and not overlapping like they were. Okay, so anyway, I'll grade the dichotomous keys and give those back to you on Thursday. Any questions? So let's go ahead and turn these in. And if you have, if you want to, if you're okay with what you got, hand them to me. We'll take a short break. I want to catch up on our lecture exam.